Today, let us take up the topic generation and conduction of nerve impulse. For the first time, generation and conduction of nerve impulse was studied by two scientists by name Hodgkin and Huxley. And for their work, they have received the Nobel Prize for Medicine or in Physiology in the year 1963. They have chosen the largest invertebrate animal for their work, and that animal is giant squid or Archaeoteuthis. Archaeoteuthis has very thick neurons or nerves. So they could easily insert an electrode to find out the voltage inside the neuron or the nerve. When they have inserted an electrode into the nerve, they found the voltage was minus 90 or 70 millivolts inside, or from minus 70 to minus 95 millivolts. So the membrane, neural membrane, is negatively charged below and positively charged on the surface. This is called resting potential. So when a nerve is not conducting any impulse, it is in a state of rest, and during rest it has potential, and that potential is known as resting potential. So a resting nerve or neuron has a resting potential of minus 70 to minus 95, and it is always measured from inside. Then, how this polarity is maintained? Further, there are three reasons. The fluid present surrounding the axon or nerve is known as extracellular fluid or tissue fluid. Inside the axon, there is a fluid known as intracellular fluid or neuroplasm. There are two types of ions in both extracellular fluid and intracellular fluid. These are sodium and potassium. They are not equally distributed, though they are present in both ECF and ICF. The sodium ion concentration is very high in extracellular fluid, but very low in the intracellular fluid. Just opposing this, the potassium ion concentration is very high inside the neuron, but it is concentration low in the extracellular fluid. That is one main difference. Then, the reason for this are two factors. The first factor is sodium-potassium exchange pump. The sodium-potassium exchange pump works against the concentration of a substance. So it requires energy, and that energy comes from ATP, adenosine triphosphate. So the sodium-potassium pump, what it does? It expels 3 sodium ions from intracellular fluid into the extracellular fluid and pumps in 2 potassium ions from extracellular fluid into the intracellular fluid. That means there is a disparity. It is removing 3 positively charged sodium ions from neuroplasm and bringing in only 2 positively charged potassium ions. This is one reason for maintaining negativity inside. The second point is about the presence of leaky potassium channels. These leaky potassium channels, they allow outward movement of potassium ions from exoplasm into the extracellular fluid. Sodium ions cannot escape like that. They don't have any leaky channels, whereas the potassium ions will be leaking out continuously from inside to the outside. Then, in the exoplasm, there are a large number of negatively charged proteins. So all these factors, sodium potassium pump, leaky potassium channels, and the presence of negatively charged proteins contribute for the negativity inside the membrane. That is the state of resting potential. Then, how we stimulate a nerve? To stimulate a nerve, we have to apply a stimulus. You know what is stimulus? Stimulus means change in the environment. We have to bring out a change in the environment, which may be external or internal. And the stimuli are various types, like heat, electricity, cold, pleasure, light, smell, etc. So when you apply a stimulus, action potentials are generated or nerve impulses are generated, and they travel along the length of the axon. Now let us see how the action potentials are generated. Now let us take up this axon, which is non-myelinated. I have identified few points A, B, C, D like that. On the surface of the membrane, it is positively charged. Along the inner surface of the membrane, it is negatively charged. Now, I have given a stimulus at point A. Then what happens? If the strength of the stimulus is below the threshold level, no response occurs. If the strength of the stimulus is above the threshold level, a nerve fiber responds completely, not partially. Whenever the strength of the stimulus exceeds the threshold level, the nerve responds completely. This is known as all or none law. All or none law. That means if the strength is below threshold stimulus, no response occurs. If the strength is above the threshold level, it responds completely, totally. So now, let us see that we have applied a stimulus of threshold strength. Then what happens? In the neurolemma, there are some channels known as voltage-gated channels for both sodium ions and potassium ions. They are called voltage-gated channels. When you apply a stimulus at point A, first the voltage-gated channels of sodium ions open, and sodium ions rush in from outside to inside at the point A. Because of inward movement of sodium ions or influx of sodium ions, what happens? The polarity at point A is reversed. Polarity at point A is reversed. Outside it becomes negative and inside becomes positive. Look at the point B. At point B, outside it is positive, inside negative. We know that electricity moves from positive pole to the positive end to the negative end, and thus it completes the circuit. So, electricity flows from point A to B inside and B to A on the outside, here and here. So, thus an electrical circuit is completed. When the electrical circuit is completed, what happens? Next, at point B, depolarization occurs. So first depolarization has occurred at point A when apply stimulus. How the depolarization has occurred? Due to influx of sodium ions from outside to inside. This is depolarization. So depolarization is due to influx of sodium ions. Now, depolarization occurs at B because of the flow of electricity. And This means there is influx of sodium ions at point B from outside. As the depolarization is taking place at point B, repolarization takes place at point A simultaneously. So, both depolarization at point B and repolarization at point A will be taking place simultaneously. So, at point A, the polarity is again reversed. Inside becomes negative and outside positive, just like as in resting potential. Then what happens between point B and C? Again, electricity flows from this positive charge point to negative point, from this positive point to negative point. So another electrical circuit is completed between point B and C. And next, at point C, the neutral undergoes depolarization, and at point B, the neutral undergoes repolarization. And these processes continue throughout the length of the axon. As a result, the action potential or impulse generated at point A first moves to point B, then C, D, like that. It travels the entire length of the axon. This can be represented in a graphic fashion like this. So this is the resting potential. Let us say minus 70 millivolts is the resting potential. So the nerve fiber is in a state of resting potential and a stimulus is given at this point. When a stimulus is given at this point, what happens? Voltage gated sodium ions. Sodium ion voltage gated channels open and sodium ions rush into the exoplasm. As a result, what happens? The negativity decreases from minus 70 to 0, then it reaches a level of plus 45 millivolts. That means the polarity is reversed from minus to plus. This is called spike. So, an action potential is generated and it has reached a level of plus 45 millivolts. Once it reaches this level, what happens? Repolarization takes place. Depolarization takes place. Here what happens? At this level, the voltage gated sodium ions suddenly close and they no longer allow the sodium ions from outside to inside. Then the potassium gates open, or voltage gated potassium channels open, and potassium opens, they rush out. They rush out from here to here. Because of the outflux or rushing out of the potassium ions, repolarization occurs. So, depolarization is due to influx of sodium ions and repolarization is due to outflux of potassium ions. Outflux of potassium ions. But at the end of repolarization, that is minus 70 millivolts, still the potassium gates are open. They are called lazy gates. They are very slow in closing. As there is delay in closing down of the voltage gated potassium channels, what happens? The negativity doesn't remain at 70 millivolts and it goes further up to 90 millivolts, minus 90 millivolts. This 70 to 90 millivolts difference is known as hyperpolarization. Hyperpolarization. After 2 or 3 milliseconds, again the negativity reaches minus 70 millivolts. The resting state is reached. In this way, action potential generated and transmitted along the axon. Now let us come to the myelinated axons. Transmission along myelinated axons is also known as saltatory transmission. It is very fast. Saltation means jumping or leaping from point to point. Here what happens is the Schwann cells, these are Schwann cells having myelin sheath, are arranged one after one throughout the length of the axon. Between two successive Schwann cells, there is a narrow gap where the neurolemma is exposed or naked. These points are known as nodes of Ranvier. These are nodes of Ranvier. At the nodes of Ranvier, the electrical charge is positive outside and negative inside. That means the nerve fiber in a polarized state. If you apply a stimulus at point A, let us say here we have applied a stimulus. Then what happens? At point A, the polarity is reversed. It means it has undergone depolarization. Outside it is negatively charged and inside positively charged. At point B, outside positively charged and inside negatively charged. Now electricity moves from point A to B and B to A like this. As the electrical circuit is completed, at point B, the neurolemma is stimulated and this point undergoes depolarization, just like here. As depolarization is taking place at point B, repolarization occurs at point A. That means the action potential generated at point A has jumped to point B. It cannot travel continuously throughout the length of the neurolemma. Here, the action potentials travel continuously throughout the length of the axolemma. But here there is no such possibility because of the presence of myelin sheath. So, the action potential generated at point A jumps from A to B, like this, from B to C, C to D, like that. It travels by jumping from node to node. It is called saltatory transmission and it is very rapid. The saltatory transmission is 5 to 50 times more faster than these non-myelinated axons. They are so fast. So in our central nervous system, we find myelinated axons. So in this way, action potentials are generated when you apply a stimulus, and these action potentials travel all along the length of the axon. This type of transmission is known as electrical transmission. So, the transmission of the impulse along the length of the axon is electrical. Then, we know that the neurons are arranged one after one and they are not connected directly. Neurons have no direct contact between them. Between two successive neurons, this is one neuron, this is another neuron, there is always a gap. It's called synaptic cleft. Synaptic cleft. So this is the axon. This is the axon terminal. The axon terminals, they swell into button-like structures known as synaptic knobs. Inside the synaptic knobs, there are synaptic vesicles. Inside the synaptic vesicles, there is a chemical substance known as neurotransmitter. Neurotransmitter. This is the synapse. The neuron present in front of the synapse is known as presynaptic neuron. This is presynaptic neuron, and this is called postsynaptic neuron. The neuron present after the synaptic cleft. So between the presynaptic neuron and postsynaptic neuron, there is a gap called synaptic cleft. Then this membrane of this neuron is known as presynaptic membrane. Presynaptic membrane, this is the postsynaptic membrane, and they are separated by a synaptic cleft. On the postsynaptic membrane, there are some protein receptors. When a nerve impulse or action potentials arrives at the synapse, the synaptic vesicles get attached to the presynaptic membrane and release the neurotransmitter into the synaptic cleft. So this is the neurotransmitter released into the synaptic cleft. The neurotransmitter acts upon the protein receptors present on the postsynaptic membrane. Same thing happens when we apply a stimulus. Sodium voltage gated channels open and sodium ions rush in, and the polarity is reversed in the case of postsynaptic membrane, and the action potentials are generated. These action potentials then travel all along the length of the dendrite. They reach the cell body, then they pass on to the axon, and again reach their terminal end. Like this, transmission across the synapse is chemical. So, up to here, it is electrical transmission. But across the synaptic cleft, it is chemical. So, if the synaptic cleft is present, in that nerve, the transmission is described as electrochemical, because partly it is electrical transmission and partly chemical transmission. So that in this way, the impulses are passed on from one neuron to the next neuron. While crossing the synapse, slight delay occurs in the transmission of the nerve impulse. This is known as synaptic delay. While crossing the synaptic cleft, some delay occurs, and this delay is known as synaptic delay. So that is about synaptic transmission. Then, these synapses are again two types: electrical synapses and chemical synapses. What I have described now is the chemical synapse. Here, the width of the synaptic cleft is about 20 to 30 nanometers. 1 nanometer means 10 angstroms. So, in the case of chemical transmission, the width of the synaptic cleft is 20 to 30 nanometers or 200 to 300 angstroms. And here, neurotransmitter is required for crossing the synaptic cleft, for the action potentials to cross the synaptic cleft. Then coming to the electrical synapses, the synaptic cleft is very narrow, only about 0.2 nanometers, very narrow. That means almost that very close, very close. So what happens? The action potentials jump from presynaptic membrane to postsynaptic membrane. It requires no neurotransmitter, and the action potentials actually jump. Because they jump from here to here, the transmission across the synapse is electrical. So, the transmission along the entire length is only electrical. Chemical neurotransmitters are not involved. So, in the case of electrical transmission, electrical synapses, the transmission is only electrical. And here, in the case of electrical synapses, there is no synaptic delay. There is no synaptic delay and they travel very fast across the synapse. Then these electrical synapses resemble gap junctions. Gap junctions we are found in the cardiac muscle and other cells. But these electrical synapses are very rare, whereas the chemical synapses are common. There is safety mechanism in the chemical synapses, in the case of chemical synapses, but that is absent here in the electrical synapses. The electrical synapses can travel in both the directions, forwards and backwards. Whereas the chemical in the case of electrical synapses, the impulses travel in both the directions. In the case of chemical synapses, the impulses travel only one direction, like this. They can't travel backwards because of the involvement of neurotransmitter substances. So, the direction of transmission in the case of electrical synapses is bidirectional, and in the case of chemical synapses, it is unidirectional from dendrites to axon. Again, then the next dendrite, axon, like that. Then, coming to the neurotransmitters, these are again two types. Excitatory neurotransmitters and inhibitory neurotransmitters. The excitatory neurotransmitters excite or stimulate the postsynaptic membrane, and as a result, the action potentials are generated and they are transmitted. That means they create impulses in the next neuron. Examples for excitatory transmitters are acetylcholine, glutamine, epinephrine, norepinephrine, etc. Then we have studied about drugs, cocaine, heroin. They are also excitatory neurotransmitters. Coming to the other type, inhibitory neurotransmitters, they inhibit transmission of impulse from presynaptic neuron to the postsynaptic neuron. They block the movement. They inhibit the activity of the action potentials. So, the impulse is stopped here. It cannot proceed to the next neuron. Communication is cut off. Examples for inhibitory transmitters are GABA, gamma amino butyric acid, serotonin. It is also used as antidepressant drug. Glycine. Then morphine, codeine, fentanyl. We have studied about morphine, opioid. These are all inhibitory neurotransmitters. So that is, in a nutshell, that is about generation and conduction of nerve impulse or action potentials. Thank you.